Imagine for just a second that you are finishing up this incredible multi-course gourmet meal at some high-end restaurant.
Oh, yeah, the kind where you can barely afford the appetizers.
Exactly. And you've just been served dessert. So maybe it's a perfectly caramelized creme brulee
Yep.
or like a really rich artisanal scoop of ice cream.
Sounds amazing.
Right. It is absolutely delicious. It's infused with this really intense, sweet, familiar flavor that tastes exactly like premium dark amber maple syrup.
Okay, I'm with you.
So you eat it, you pay the bill, you go home. But then a few hours later, you go to the gym, you start to work out, you start to sweat, and you suddenly realize something super weird.
Your own sweat smells exactly like that dessert.
Yes. Your sweat, your urine, your skin, you are literally radiating the intense sweet scent of maple syrup.
It is a genuinely startling experience, especially if you aren't anticipating it. I mean, your entire biological exhaust system has basically been temporarily rewritten to smell like a pancake breakfast.
It is wild. And the twist here is what actually caused it. Because that gourmet ingredient responsible for the incredible dessert and, your incredibly sweet-smelling sweat, it isn't maple syrup at all.
Right. Not even close.
It is vegetrum. But what makes this completely bizarre to me is how history has actually treated this exact same organism.
Oh, the history is the best part.
Yeah, because if you serve this sweet-smelling mushroom to a 19th century European, they wouldn't think of breakfast. They would be utterly repulsed.
Yeah.
Because to them, it was known simply as the bedbug mushroom.
Which is arguably one of the most fascinating contradictions in the entire natural world.
Right.
Yeah, I mean, how a single organism can span the sensory gap between a luxury dessert and a literal parasitic insect infestation.
So welcome to this deep dive. Today, we are exploring the totally unique chemistry, the fascinating facts, and the completely bizarre history of Lactarius camphoratus.
Widely known today as the Curry Milkcap or the candy cap.
Right. And the reason we are spending an entire conversation on this one specific tiny forest fungus is that it acts as this perfect microscopic lens for so many different fields of study.
It really does. It sits at the exact intersection of human sociology, volatile chemistry, underground ecological warfare, and even modern medical science.
Yeah. Which just proves how little we actually understand about the ground beneath our feet.
Exactly. We often look at a forest floor and, we just see dirt, leaves, maybe a few mushrooms popping up after a rainstorm.
Just sitting there passively.
Right, we think of it as static. But this organism demonstrates that the soil is a highly active chemical laboratory. I mean, it's producing compounds that can literally alter human metabolism.
So to understand how this mushroom works and how it manages to manipulate our senses so drastically, we kind of have to look at how humans have historically tried to classify it.
And honestly, the early attempts were a complete mess.
A total mess. The scientific recognition of this mushroom goes back to 1792 in France with a mycologist named Jean-Baptiste François Pierre Bouillard.
Yeah, Bouillard was operating in an era of really intensive, almost frantic botanical categorization in Europe.
They were basically trying to build a library of life from scratch.
Precisely. He provided the first formal description of this fungus, and he gave it the scientific name Agaricus camphoratus.
Now, throwing it into the Agaricus bucket kind of made sense at the time because in the late 18th century, early botanists basically used that genus as a massive dumping ground.
Oh, absolutely. If a mushroom had gills on the underside of its cap, they just slapped the name Agaricus on it and moved on.
Right. It wasn't until much later that they started looking at internal structures to separate them all out. But the second part of that name, the camphoratus part, that is what really catches my attention.
Because of the camphor connection?
Yeah. Bouillard named it that because he thought it smelled like camphor. But what exactly was camphor to someone living in 1792?
Well, camphor is a highly pungent bicyclic monoterpene ketone. It was originally derived from the wood of the camphor laurel tree in Asia.
Okay, so a very strong smell.
Very strong. Today, you might recognize that distinct, cooling, kind of medicinal smell if you open a jar of Vicks Vapor Rub or like a tin of Tiger Balm.
Oh, okay.
Yeah.
Yeah, I know that smell.
But back in the 18th and 19th centuries, camphor was ubiquitous. It was used in everything.
Like what?
People packed their clothes in it as a moth repellent. Apothecaries used it as an anti-inflammatory medicine. It was even a key ingredient in early plastics and explosives.
Wow, so it was basically the default strong medicinal smell of the entire era.
Exactly.
But if you walk into the woods today, you pick up a fresh Curry Milkcap and you smell it, you don't get mothballs. You definitely don't get medicinal ointment.
Not at all.
So modern mycologists seem incredibly puzzled by Bouillard's choice of the word camphoratus.
Right, they're puzzled because the fresh mushroom simply doesn't possess that sharp, cooling terpene profile.
So why did he name it that?
Well, the prevailing theory today is that Bulliard wasn't actually out in the forest taking notes on fresh, living mushrooms.
Oh, he was in a lab.
Yeah, he was likely working in an herbarium, studying dried specimens that had just been sitting in cabinets.
Oh.
And as we'll discuss in a bit, the entire chemical profile of this mushroom completely transforms when it loses its moisture.
Okay, that makes total sense.
Mm.
He's sitting in a dusty room smelling a dried husk. So if we fast forward to 1838, the foundational Swedish mycologist Elias Magnus Fries steps in.
Right, and he completely reorganizes things.
He realizes the agaricus dumping ground is totally useless, and he moves this species into its current home, which is the genus Lactarius.
And that was a really brilliant piece of taxonomic correction.
Because he was looking at the actual anatomy, right?
Exactly. Fries was looking at the anatomy of the organism itself. The name Lactarius actually comes from the Latin word for milk.
Okay.
Fries realized that this mushroom, along with its close relatives, possesses specialized cellular networks. They're called lactiferous hyphae.
So what does that actually look like inside the mushroom? Are there just little veins of milk running through the stem?
Essentially, yes. They're these specialized tube-like structures that run throughout the flesh of the fungus, totally separate from the structural cells,
Wow.
and they are highly pressurized. So when the physical body of the mushroom is, say, bitten by an insect or sliced by a knife, that pressure is released.
Like popping a tire?
Yeah, and these tubes immediately exude a milky liquid latex to basically seal the wound.
Okay, so Fries correctly identifies that it bleeds milk, renames it Lactarius camphoratus, and puts it in the right family.
Yep.
But here is where the history gets completely unhinged.
Oh, the bedbugs.
Yes. If you look at the early English language field guides and mycological journals, they do not call it the Curry Milkcap. They don't call it the candy cat,
No, they call it the Cimicin or the bedbug Lactarius.
which is just a phenomenal detail. And it traces right back to Elias Magnus Fries himself, right?
It does. When he was documenting the physical traits of the mushroom, he made a very specific note about its smell. In Latin, he wrote odor vago in sicis cimetino.
Which translates to a vague odor, but when dry, like bedbugs. Exactly.
I cannot imagine walking through a beautiful pine forest, picking up a mushroom, and thinking, yes, the distinct aroma of a parasite-infested mattress.
It's hard for us to grasp today, but to understand why Fries made that connection, we really have to look at the biology of the bedbug itself.
Cimex lectularius, right?
Yes. Bedbugs possess these specialized glands on the ventral side, the underside of their bodies, and when they are disturbed or threatened, they release a defensive secretion.
Oh, gross.
And that secretion happens to have a very heavy, musty, sweetish odor.
Okay, so there is an actual physical chemical overlap in the scent profile between the bug and the mushroom.
Exactly.
But making that connection requires a person smelling the mushroom to be intimately familiar with the smell of bedbugs, which honestly tells us a really sad amount about life in 19th century Europe.
It does, unfortunately. Before the advent of synthetic pesticides and, modern indoor plumbing, bedbugs were an incredibly common, almost inescapable part of urban and rural life.
Wow. If you walked into a crowded boarding house or an unventilated bedroom, the ambient smell of that sweet, musty, defensive secretion was everywhere. It was basically a primary cultural reference point for a heavy, sweet aroma.
It totally reminds me of how we still use a floppy disk icon on a computer to mean save.
Oh, that's a perfect comparison.
Right. An entire generation of kids growing up today has never touched a physical floppy disk, but the symbol is culturally locked in time. We just use the vocabulary of our environment.
Exactly.
So as the Industrial Revolution progressed, and as the middle class got better hygiene, bedbugs were eventually eradicated from everyday polite society.
Right.
So that olfactory vocabulary had to break down, didn't it? You can't describe a mushroom as smelling like a bedbug if literally no one knows what a bedbug smells like anymore.
Precisely. And you see a really distinct sociological shift in the literature because of that.
What happens?
Well, as the reference point of the bedbug faded from public consciousness, mycologists and foragers needed a new way to describe this intense, sweet smell. And right around the mid-20th century, as global trade routes expanded and new culinary ingredients flooded Western markets, the descriptors shifted completely.
So people started saying it smelled like curry spices or fenugreek or burnt sugar or maple syrup.
Yes, they mapped the ancient forest smell onto the new exotic spices sitting right in their kitchen cupboards.
That is fascinating. But that brings up a massive mechanical question for me.
Okay, what is it?
How does a living mushroom that smells vague or honestly like nothing at all when it's fresh suddenly turn into a curry bomb or a maple syrup factory the literal moment you dry it out?
This is where the biology shifts into pure, elegant chemistry.
I love when that happens.
The answer lies in a major breakthrough that occurred in 2012. It was led by a chemical ecologist named William Wood at Humboldt State University.
Okay.
Wood was investigating a very closely related West Coast species called Lactarius rubidus. It shares the exact same scent profile. And what his team discovered completely upended how we thought about mushroom aromas.
Because they found that the fresh mushroom doesn't actually contain this scent molecule at all, right?
Right. When the mushroom is living, growing in the damp soil, the molecule that makes it smell like maple syrup simply does not exist inside its tissues.
Why, it doesn't exist at all.
Nope. Instead, the milky latex and the cellular walls are packed with a completely odorless precursor compound. It's an aromatic lactone known as quabalactone III.
Okay, so it's just sitting there completely scentless, acting like this chemical reservoir.
Exactly. But the moment you pick it and put it in a dehydrator or even just leave it on a counter to dry, the magic happens.
The moisture leaves the mushroom.
Yes, and that moisture loss triggers a chemical cascade. Specifically, it triggers a process called hydrolysis.
Okay, break that down for me.
As the residual water within the mushroom tissue begins to evaporate and interact with the cellular environment, it violently cleaves the quabalactone III molecule.
The actual formula for this is so fascinating. You have the odorless quabalactone III, which is C6H9NO2, and then you add the water, H2O. And the water basically acts like a chemical crowbar.
That's a great way to put it.
It pries the nitrogen out of the molecule, releasing it as ammonia or NH3. And once that ammonia is stripped away, the leftover atoms instantly snap into a completely new configuration.
C6H8O3, which is the molecule Sotolon.
Sotolon.
Sotolon. Its formal chemical name is 3-hydroxy-4-fully-5-dimethyl-2,5-H, furanone.
Say that 5 times fast.
I'd rather not. But it is one of the most powerful flavor shapeshifters in all of organic chemistry.
Shapeshifter is honestly the perfect word. Because the way we perceive Sotolon completely depends on how much of it is actually hitting our nose, right?
Concentration is everything.
Yeah.
At very low concentrations, Sotolon interacts with our olfactory receptors to produce a warm, rich, incredibly sweet sensation.
Which the brain interprets as maple syrup or caramel or molasses.
Exactly. But if you increase the concentration, if you pack more of those molecules into the air,
the perception shifts aggressively.
yes. The sweet dessert notes vanish completely, and it transforms into the heavy, spicy, savory pungency of curry powder, fenugreek seeds, and lovage root.
It is exactly like the volume knob on a stereo amplifier.
How so?
Well, if you keep the volume at like the 2 or a 3, the speaker is playing this soothing, sweet acoustic melody. That's the maple syrup.
Oh, yeah.
But if you crank that exact same dial up to 11, it distorts. It becomes this aggressive, heavy metal wall of curry spice. It is the identical song, the identical molecule, but the intensity completely rewires how your brain processes it.
That is a phenomenal analogy. And this perfectly explains why Lactarius camphoratus is called the Curry Milkcap, while its cousin Lactarius rubidus is often just claimed a candy cap.
Because of the concentrations.
Right. The European and Eastern U.S. species tends to dry in a way that produces a much higher concentration of Sotolon. It pushes it right into that curry territory.
Wow.
Whereas the West Coast species tends to hover at the lower maple syrup threshold.
But wait, the chemistry gets even more complex than just the volume knob. Because there is a structural reality to Sotolon that absolutely blows my mind. The molecule actually has a handedness, chirality.
Yes, enantiomers. Sotolon is a chiral molecule, which means it exists in 2 distinct forms that are non-superimposable mirror images of each other.
Okay, what does that mean in plain English?
Think of your left and right hands. They have the exact same number of fingers, the exact same structure, but you cannot perfectly lay one on top of the other so that they match exactly. They occupy space differently.
Right. And you're telling me that a left-handed molecule and a right-handed molecule made of the exact same atoms, just flipped, actually smell completely different to us. How does our nose even know the difference?
Because the olfactory receptors inside your nasal cavity are themselves chiral structures. They act like highly specific locks.
Oh, I see.
So a left-handed molecule will fit into a specific receptor lock perfectly, turning the key and triggering a massive electrical signal to the brain.
But the right-handed one won't.
Right. The right-handed mirror image might bump into that exact same lock, but because the shape is flipped, it won't turn the key. It literally has to go find a completely different receptor to bind to.
That is wild. So with Sotolon, we have the plus S enantiomer and the minus R enantiomer.
Exactly. The S enantiomer is the one responsible for the sweet caramelic maple notes. And its binding affinity with our receptors is stunningly powerful.
How powerful.
The human nose can detect the S enantiomer at a threshold of just zero point 0.8 parts per billion.
zero point 0.8 parts per billion. That is an almost unfathomable level of sensitivity. That's like being able to smell a single drop of liquid dissolved into an entire Olympic-sized swimming pool.
It is an evolutionary marvel. Now, contrast that with its mirror image, the R enantiomer.
Okay.
When you isolate the R enantiomer, it doesn't smell sweet at all. It is described as rancid, somewhat walnut-like, and highly unpleasant.
Thanks.
Furthermore, its detection threshold is 89 parts per billion.
Wait, so it takes over 100 times more of the rancid mirror image molecule for us to even register its existence compared to the sweet molecule.
Exactly, which is incredibly fortunate for anyone cooking with this mushroom.
No kidding.
The fact that the Curry Milkcap is highly prized culinarily suggests two things. Either the mushroom's internal enzymes heavily favor the production of the sweet S enantiomer during hydrolysis, or the rancid R enantiomer's high detection threshold simply renders it invisible beneath the overwhelming power of the sweet side.
Okay, this deep dive into molecular structure perfectly explains the hook we started with, the gourmet dessert and the maple-scented sweat at the gym.
Hmm.
How exactly does eating Sotolon hijack human sweat glands?
It's widely known in the literature as the maple sweat phenomenon.
Maple sweat.
Yeah. When you ingest significant quantities of Lactarius camphoratus, you are basically flooding your system with Sotolon. And unlike many other complex flavor molecules, Sotolon is incredibly robust.
What do you mean by robust?
It passes through the harsh acid of the human stomach and the intense enzymatic breakdown of the liver and digestive tract relatively unchanged.
It just survives the whole gauntlet.
It does. It enters the bloodstream completely intact, and because the body cannot easily break it down for energy, it has to excrete it somehow.
Right.
So the Sotolon is pushed out through the renal system into your urine and pushed out through the apocrine and eccrine glands right into your sweat.
You literally become a biological conduit for the mushroom's aroma.
Exactly. It's very similar to how sulfur compounds in garlic seep from your pores, just, significantly more pleasant.
Yeah, much rather smell like pancakes than garlic.
Oh.
But there is a medical angle to this that is genuinely shocking. Because smelling like maple syrup isn't just a quirky side effect of eating a mushroom. It is also the primary diagnostic symptom of a very serious human disease.
Yes. Sotolon is heavily studied in medical literature because it is the exact same molecule responsible for the odor in a genetic metabolic disorder called maple syrup urine disease, or MSUD.
I have to admit, MSUD sounds almost made up. What is actually happening in the body to cause that?
Well, MSUD is a rare inherited genetic disorder. In a healthy human body, we consume proteins, we break them down into amino acids, and we use specific enzymes to process them.
Right.
But individuals with MSUD are born lacking the specific enzymatic machinery required to break down three specific branched-chain amino acids: leucine, isoleucine, and valine.
So because they can't break them down, these amino acids just start backing up in the system like a clogged pipe.
Exactly. The amino acids and their toxic byproducts accumulate in the blood and tissues, which can lead to severe neurological damage if it's not managed immediately with a specialized diet.
Oh, wow.
But the key here is the byproduct. As the body desperately tries and fails to metabolize these compounds, it undergoes abnormal chemical reactions.
Okay.
And one of the direct results of that abnormal metabolism is the spontaneous synthesis of Sotolon.
So when a pediatrician walks into a room and notices that a newborn's diaper smells intensely of maple syrup, they are literally smelling the exact same molecule that a woodland fungus produces. The broken human metabolism accidentally manufactures the mushroom's chemical signature.
It is a profound reminder of how universal these chemical building blocks are across vastly different kingdoms of life. I mean, the exact same arrangement of carbon, hydrogen, and oxygen can be a defense mechanism in a fungus, a flavor enhancer in a kitchen, or a red flag for a lethal genetic disorder in a human.
It is absolutely stunning. Okay, so we know this mushroom is a marvel of chemistry. We know what it does to the body. But this brings us to a very practical, boots-on-the-ground problem.
Identifying it.
Yes. If someone listening wants to go out into the woods and find Lactarius camphoratus, how do they do it safely? Because as we've learned, you can't just walk around sniffing fresh mushrooms. The quabalactone, the third, is odorless.
Right.
And the forest is absolutely loaded with dangerous imposters.
It is. Identifying this species in the wild is an exercise in careful macromorphology. You really have to look at the physical architecture of the mushroom, and you have to ignore scent completely until the specimen is safely back home and dried out.
Let's pull out the visual and tactile traits. If I'm walking through the woods, what am I looking for? First off, it's not huge, right? The cap is typically only 3 to 6 centimeters in diameter.
Yes, it is a relatively diminutive species. When it is young, the cap is convex, so it's curving downward like a dome.
Okay.
But as it matures and expands, the center of the cap begins to sink, creating a central depression.
And right in the middle of that depression, there's often a tiny, sharp little point. Mycologists call it a papilla or an umbo, right?
Yes, an umbo. Color is also a useful, though somewhat variable metric. You're looking for a dark reddish-brown to a deep rusty orange.
But that can change, right?
It can. Environmental factors like heavy rain or prolonged sunlight can bleach the pigments, causing older specimens to fade significantly.
Got it. What about underneath?
The gills underneath are typically a pinkish cream color that darkens to a pale cinnamon as the spores mature. And those gills are slightly subdecurrent, which just means they run a little bit down the stem.
But the texture is where things get really interesting to me. Because you don't just look at a mushroom, you have to feel how it breaks.
Oh, absolutely. The structural integrity of the Lactarius genus is totally unique. The internal flesh, known as the context, is pale cinnamon to whitish. It is very hard, but extremely brittle.
Brittle how?
Well, if you take the stem of a curry milkcap and you snap it in half, it doesn't bend, and it doesn't leave stringy, fibrous threads like you'd see if you tore a grocery store button mushroom.
Okay.
It fractures cleanly, almost exactly like snapping a piece of classroom chalk.
Wait, why does it break like chalk?
Because of the shape of its cells. Many mushrooms are built entirely from long thread-like hyphae that are woven together, but Lactarius species incorporate these clusters of spherical cells called spherocysts right into their tissue.
So round cells.
Yeah, these round cells create structural weak points, which causes the flesh to snap cleanly rather than tear.
Fascinating. But the ultimate field test, the absolute deal breaker you have to perform if you think you found one, is the latex test.
This is the critical diagnostic marker. When you take your knife and slice across those subdecurrent gills, or snap the cap in half, you observe the milk.
And what does it look like?
Initially, the latex exuded from those pressurized hyphae will be an opaque, milky white. But, and this is the key, that white milk will rapidly change consistency.
Rapidly change to what?
It becomes thin, watery, and almost clear.
It's described in the literature as whey-like someone took a drop of skim milk and mixed it with a cup of water.
Exactly. That rapid transition from an opaque white drop to a thin, watery smear is the absolute hallmark of Lactarius camphoratus.
And what about tasting it? Can you do a taste test in the field without swallowing?
You can perform a tiny chew and spit test with the latex, but with extreme caution. The latex of the curry milkcap is mild. It might be slightly bitter or even vaguely sweet on the tip of the tongue.
What are we trying to avoid?
Crucially, it never possesses a burning, peppery acridity. If it burns your mouth, spit it out immediately because you have the wrong mushroom.
Okay, so you have a small reddish-brown mushroom, chalky stem, whey-like milk, mild taste. But if you want to be 100% sure, you bring it back to a workbench and you run the chemistry.
Yeah.
What kind of chemical reagents do mycologists drop on these things?
The standard tests involve pretty basic household or laboratory chemicals. If you apply a drop of potassium hydroxide or KOH directly to the surface of the cap, the specific pigments in Lactarius camphoratus will undergo a reaction and instantly turn olive green.
Oh, cool.
And if you apply a crystal of ferrous sulfate to the internal stem tissue, it will oxidize and turn a color ranging from salmon pink to blue-green, depending heavily on the moisture and freshness of the tissue.
And then there is the microscopic level. This is my favorite part of taxonomy. It's literally like dusting for fingerprints at a crime scene.
It really is.
You take the cap, you place it on a glass slide overnight to drop its spores, and then you look at that pale yellow spore print under high magnification.
Under a high-powered microscope, you are looking at spores that measure roughly 7.0 to 8.5 micrometers long by 6.0 to 7.5 micrometers wide.
So tiny.
Very tiny. They're broadly ellipsoidal, so almost spherical but slightly elongated like a very plump football. But the true fingerprint is the ornamentation on the outer shell of the spore.
The amyloid ornamentation. These are tiny microscopic bumps and ridges that chemically react with iodine, turning a dark blue-black color so you can see them clearly.
Correct. The spores of the Curry milkcap are covered in isolated warts and scattered ridges that stick up about half a micrometer. But the absolute key identifier, the fingerprint, is that these ridges do not connect with each other to form a closed, grid-like network.
And in mycology, a grid pattern is called a reticulum.
Yes. Lactarius camphoratus is defined by having non-reticulate spores. The ridges just kind of wander off and stop.
Now, I have to play devil's advocate here. Why on earth do we need to look at half-micrometer spore ridges under a laboratory microscope if the smell of the dried mushroom is so overwhelmingly unique? Can't a forager just dry the mushrooms they find, wait to see if the kitchen smells like curry, and throw away the ones that don't?
You could, but you are playing a very dangerous game with your liver.
Fair point.
Relying solely on the volatile scent profile is incredibly risky due to the existence of localized look-alike species that share the same color palette and in some cases share the exact same Sotolon chemistry.
Let's talk about those imposters because they are fascinating in their own right. First, there's the broader candy cap complex in North America.
Right. When foragers in North America talk about candy caps, they are generally referring to three closely related species. You have Lactarius camphoratus in the eastern U.S. and Europe, but if you go to the western United States, specifically the coastal pine forests and the Cascade Mountains, you find Lactarius rubidus. And also in the east, there is a third species, Lactarius fragilis.
And all three of these share the quabalactone 3 to Sotolon pathway. They all smell like maple or curry when dried.
They do, and all three are considered choice edibles. However, if you put them under a microscope, L. rubidus and L. fragilis differ from our primary subject because they do have reticulate spores. Their spore ridges connect to form that net-like grid.
But getting those mixed up isn't really dangerous.
Yeah.
Right? The real danger lies in the toxic lookalikes.
Exactly. Like the European species, Lactarius helvis.
Tell me about Helvis.
Lactarius helvis is the perfect example of why smell alone is a trap. It is a slightly larger mushroom, heavily found in swampy European pine woods, and it also heavily synthesizes Sotolon.
Oh, no. So it smells the same.
Yep. When it dries, it produces a massive pungent aroma of fenugreek and maple. If you were blindly relying on scent, you would be absolutely certain you had a Curry milkcap.
But eating it is a terrible idea.
It is mildly toxic. Consuming Lactarius helvis will cause severe gastrointestinal distress. It contains entirely different secondary metabolites that really irritate the human gut.
Okay, so how do you tell them apart?
You can distinguish it easily in the field if you pay attention to the milk. Its latex is completely clear and watery right from the exact moment you cut it. It doesn't start white in transition, it is just clear liquid.
Okay, good to know. And then there is the rufous milkcap, Lactarius rufus.
Right. Lactarius rufus shares the dark red brick color of our subject. But if you slice its gills, the latex that bleeds out is persistently thick and white.
It never turns watery.
It never turns watery. And if you do the taste test, you will know instantly. The milk is violently acrid. It will severely burn your tongue and throat.
Yikes.
It is classified as poisonous, though in certain regions of Eastern Europe, they do utilize a traditional method of extensive, repeated parboiling to leach out the toxins before eating it. But it is absolutely not something a casual forager should ever attempt.
So the spore fingerprint, the milk consistency, the lack of a burning taste, all of these elements really stack together to create a safe identification.
Exactly.
Okay, so we've successfully and safely identified the mushroom. We understand its shape-shifting chemistry. Now, I want to pivot to how this organism actually lives and survives in the wild. Because the Curry milkcap isn't just a passive little lump sitting in the dirt waiting to be picked.
Not at all.
It is locked into a highly active, dynamic relationship with the entire forest ecosystem. It's both an indispensable partner and, kind of tragically, a victim.
The ecological footprint of this fungus is massive. Lactarius camphoratus is a cosmopolitan species. That means its genetic lineage has successfully colonized vast swaths of the northern hemisphere, spanning across Europe, into Asia, and throughout the eastern half of North America.
And its success is entirely due to its primary ecological role, right? It is an ectomycorrhizal symbiont.
Right.
Ectomycorrhizal, let's pull that apart. What does it actually mean for a fungus to be an ectomycorrhizal symbiont?
It means it has evolved to form a mandatory, mutually beneficial physical connection with the root systems of specific trees.
Like which ones?
In the case of the Curry milkcap, it primarily targets conifers like spruce and pine, though it can also adapt to certain hardwoods like oak.
Okay.
The main body of the fungus isn't the mushroom we see. It is a vast, microscopic underground network of biological threads called mycelium. And this mycelium physically wraps itself around the microscopic root tips of the host tree, forming a protective sheath.
I always think of this underground network as an internet service provider, an ISP for trees.
That's a great analogy.
Because the tree is towering above the canopy, acting like a giant solar panel. It's photosynthesizing, pulling carbon dioxide out of the air, and manufacturing massive amounts of liquid sugars.
Right.
But the tree has a physical limitation. Its roots are thick and clunky. It struggles to extract enough raw minerals, phosphorus, and water from the tight, microscopic crevices of the soil.
Exactly, and the fungus has the exact opposite problem. The fungal mycelium is composed of threads that are only a single cell wide, so it is a master at mining the soil.
It gets into all the tiny cracks.
Yes, it can secrete enzymes to dissolve rocks, releasing trapped phosphorus and nitrogen, and it acts like a massive sponge for water. But the fungus has no access to sunlight. It cannot manufacture its own carbon-based food.
So they set up a trade agreement.
The ectomycorrhizal connection is the trading post. The fungus pumps the water and dissolved minerals into the tree's roots, essentially acting as a massively expanded secondary root system.
And in return,
In exchange, the tree pumps up to 20% of the liquid sugars it creates straight down into the roots and feeds the fungus. They are entirely reliant on each other.
it's this beautiful, invisible partnership that literally keeps the forest alive. And the scale of this partnership is staggering. Did you know that in Estonia, the Curry Milkcap is so incredibly common, and its mycorrhizal network is considered so vital to the health of their massive lichen-rich pine forests, that the Estonian Museum of Natural History officially named it the Fungus of the Year in 2024.
Which is just a brilliant way to acknowledge that the health of the towering pines is entirely dependent on the microscopic chemistry happening beneath the moss.
But even the most sophisticated networks get hacked.
Yes, they do.
And this brings us to a truly dark, fascinating twist in the ecological record. Because there are field observations, specifically detailed reports from the Little Thicket Nature Sanctuary in Cleveland, Texas, that document what happens when the Curry Milkcap gets attacked.
Yes. The Texas observations highlight the brutal reality of interfungal parasitism.
Fungi attacking fungi.
Right. Lactarius camphoratus is highly susceptible to attack by another fungus. It's a parasitic mold known as Hypomyces camphoratus, or historically Pechiella camphoratus.
How does it even attack it?
Well, the spores of the Hypomyces parasite land on the developing fruiting body of the Curry milkcap. As the parasite's mycelium grows, it physically invades the cellular structure of its host.
Oh, wow.
It consumes the host's tissue, entirely hijacking its biological processes. As the infection progresses, the beautiful reddish-brown cap and the gills become completely deformed, transforming into a swollen, pale, sterile mass of parasitic tissue.
It literally eats the mushroom from the inside out while it's still attached to the tree network.
It does.
But the most crucial detail here, tying all of this back to our earlier discussion about maple syrup in chemistry, is what this parasite does to the host's scent.
This is the truly remarkable part of the infection. When the Curry milkcap is heavily infected by the Hypomyces parasite, the development of the mushroom's characteristic aroma is completely suppressed.
Wait, really?
Yes, the infected mushroom dries out, but it doesn't smell like curry and it doesn't smell like maple.
The hacker literally turns off the chemical factory.
It does. This biological stressor proves that a parasite can completely override the host organism's secondary metabolic pathways.
How?
Well, the production of quabalactone III takes biological energy. The parasite, acting as a metabolic dictator, forcefully redirects the Curry milkcap cellular energy away from producing that useless scent precursor and forces it to use that energy solely to fuel the growth of the parasite.
That is just incredible. The fact that a parasite can flip a switch and turn off the host's metabolism brings us perfectly to the next major revelation. Because the quabalactone III scent precursor isn't the only chemical this mushroom manufactures.
Not by a long shot.
Beyond its shape-shifting scent and its underground tree network, the Curry milkcap is an absolute factory for highly complex bioactive compounds. When we look at the pharmacological data, we find an entire chemical arsenal designed for war.
Because the sweet scent is ultimately just a byproduct of drying. While the mushroom is alive, it is constantly defending itself against slugs, nematodes, insect larvae, and bacterial infections.
It's a battlefield down there.
And the way the Lactarius genus deploys its primary chemical defense is ingenious. It operates exactly like a binary weapon system.
A binary weapon system.
Yeah.
Okay, I love this concept, but explain how a mushroom weaponizes itself.
So, in many species within this genus, the milky latex we discussed earlier is more than just a sealant. It is the delivery mechanism for a weapon.
Okay.
The latex contains a completely inactive, harmless precursor chemical. Floating alongside it in the cellular fluid are specific, highly reactive enzymes. But as long as the mushroom is healthy and intact, these two components are strictly segregated by fragile cellular membranes or kept apart as a stable emulsion.
It's exactly like a modern glow stick.
Oh, perfect.
Right. When you hold a glow stick, you have one chemical floating freely in the plastic tube, and a second, highly reactive chemical trapped inside a tiny, fragile glass vial floating in the middle. It does absolutely nothing until you bend the plastic, snap the internal glass, and force the two chemicals to mix.
That is a flawless analogy. The moment a hungry slug bites into the gills of the mushroom, or an insect larva burrows into the stem, the physical trauma ruptures those internal cellular walls.
The glass breaks.
Yes. The inactive precursor and the enzyme are violently forced together. Within mere seconds of the injury, the enzyme catalyzes the precursor, instantly converting it into highly pungent, acutely toxic dialdehyde compounds.
So the slug takes a bite of a perfectly harmless mushroom, and before it can even swallow, the mouthful of tissue chemically detonates into a toxic poison.
Precisely. This rapid, localized chemical response effectively burns the mouthparts of herbivores, instantly deterring predation and ensuring the mushroom survives long enough to drop its spores.
That is evolutionary genius. But the defensive dialdehydes are just the frontline infantry. Modern pharmacological research has dug much deeper into the molecular structure of Lactarius camphoratus.
Yes, and they've isolated an incredible array of secondary metabolites that have profound medical potential for humans.
Right. The research highlights a specific class of compounds called sesquiterpenes, specifically one named 12-hydroxycaryophylline 4-thiophos oxide.
Yes. Now, unlike Sotolon, this specific caryophylline compound is non-volatile. It doesn't evaporate into the air to create an odor.
Okay.
But within the human body, caryophylline-derived sesquiterpenoids are highly bioactive. In vitro pharmacological studies, they exhibit significant anti-inflammatory effects.
How does that actually work? If you ingest these compounds, how do they physically stop inflammation in human tissue?
They operate at the cellular signaling level. When the human body is injured or fighting an infection, immune cells release signaling proteins called cytokines to trigger inflammation, swelling, and heat.
Right.
Two of the primary culprits in chronic inflammation are interleukin-1, or IL-1, and interleukin-6, IL-6.
Okay.
The caryophylline compounds isolated from this mushroom have been shown to actively downregulate the expression of these specific cytokines.
So they basically intercept the body's signal to inflame,
Right.
reducing the tissue's inflammatory response.
Ex-
So they chemically tell the immune system to just calm down. But the medical potential doesn't stop there. The literature indicates these compounds also have serious anti-cancer potential.
Correct. Specific research on these extracts indicates they can induce apoptosis in colon cancer cell lines.
Apoptosis. For anyone unfamiliar, that is programmed cell death, right?
Yes. Healthy human cells are programmed with a biological kill switch. If they become heavily damaged or mutated, they trigger apoptosis and safely destroy themselves.
The cancer cells don't do that.
Right. Cancer cells have mutated to ignore that kill switch, allowing them to multiply uncontrollably. The sesquiterpenes from the Curry Milkcap appear capable of bypassing that mutation, forcefully flipping the kill switch back on, and causing the colon cancer cells to self-destruct.
Which effectively suppresses their ability to migrate and invade healthy tissue.
Exactly.
It's acting like a chemical assassin, targeting the rogue cells. And on top of that, it demonstrates antiviral activity, specifically showing effectiveness at neutralizing the influenza A virus, along with broad antimicrobial activity against various pathogenic diarrhea.
It is literally a pharmacy contained within a 3-centimeter cap.
And we haven't even touched on the high molecular weight polysaccharides yet.
Right. The macromolecular structure of the mushroom is equally impressive. When researchers extract the complex sugars, particularly the beta-glucans, they find highly potent biological modifiers.
Beta-glucans are well documented throughout mycological medicine for their ability to activate the human immune system and drastically reduce oxidative stress, aren't they?
They are.
I noticed the lab assays mentioned in the data, things like the DPPH and FRAP assays. These tests confirm that the mushroom's extracts are incredibly efficient at neutralizing free radicals.
And neutralizing free radicals is a massive deal. Because free radicals are essentially highly unstable rogue molecules that bounce around inside your body, tearing apart healthy DNA and cellular structures. That cellular damage is a primary driver of biological aging and the initial onset of cancer.
Wow. So by neutralizing those free radicals, the beta-glucans act as a biological shield.
Yes. Furthermore, in vitro studies testing these specific Lactarius extracts against human cancer lines are highly promising. They have been shown to induce cell cycle arrest in several aggressive lines, particularly MCF7 breast cancer, A549 lung cancer, and DU145 prostate cancer.
Cell cycle arrest. So it doesn't just kill the cell, it physically freezes it.
It halts the complex machinery required for cell division. A cancer cell's only goal is to divide and conquer.
Right.
By inducing cell cycle arrest, the mushroom extract physically stops the cancer cell from replicating its DNA and dividing into two cells. It stops the tumor's growth in its tracks.
Okay, we have anti-inflammatory, anti-cancer, and antiviral properties.
Yeah.
But there's one final major medical pathway being researched that completely blew my mind: neuroprotection.
This is arguably one of the most exciting frontiers in modern pharmacological research.
Tell me about it.
Preliminary lab studies have demonstrated that organic extracts from Lactarius camphoratus exhibit potent anticholinesterase activity. Specifically, they act as inhibitors against two crucial enzymes in the human brain:
Which are?
acetylcholinesterase, or ACE, and butyrylcholinesterase, BCHE.
Okay, let's break down the neurology here. Why does inhibiting those two enzymes matter? What exactly does that accomplish inside the brain of someone suffering from cognitive decline?
To understand the mechanism, we have to look at how neurons talk to each other. Acetylcholine is a fundamental neurotransmitter in the brain, heavily responsible for facilitating memory formation, learning, and attention.
Okay.
When a neuron fires, it releases acetylcholine into the synapse, the gap between cells, to pass the message along. Once the message is received, the brain releases enzymes, specifically ACE, to act like chemical scavengers.
Their job is to break down the acetylcholine and clear the synapse so the next message can be sent.
Yes.
So the enzymes are the brain's cleanup crew.
Exactly. But in neurodegenerative disorders like Alzheimer's disease, the brain's architecture is degrading, and there is a severe catastrophic shortage of acetylcholine being produced. The neurons are struggling to send messages.
Oh, I see.
If the ACE cleanup enzymes are allowed to operate normally, they wipe out the tiny bit of acetylcholine that is left, completely halting memory formation and cognitive function.
So if the mushroom extract inhibits the cleanup enzyme,
It creates a bottleneck. By inhibiting ACE and BCE, the natural mushroom compounds prevent the breakdown of the neurotransmitter.
so it artificially maintains a much higher concentration of acetylcholine floating in the brain synapses.
Right, which allows the damaged neurons a much better chance of successfully transmitting memory signals.
That is incredible.
It is. And this mechanism, enzyme inhibition to maintain neurotransmitter levels, is the exact primary pharmacological pathway utilized by currently prescribed synthetic Alzheimer's and dementia medications.
Wait, really?
Yes. The fact that a natural compound found in the Curry Milkcap can perform this identical, complex enzymatic inhibition is incredibly exciting for researchers searching for sustainable, plant-based neuroprotective drugs.
It's a profound shift in perspective.
Yeah.
When you start with a mushroom that makes a gym-goer smell like a pancake, and you end up exploring molecular pathways that could treat Alzheimer's and halt the division of lung cancer.
It's quite the journey.
Which naturally brings us to the final aspect of our exploration: culinary ethnomycology. With all of this intense pharmacology, this binary weapon system, this shape-shifting chiral chemistry, how do humans actually interface with this organism in daily life?
Well, the first rule of the Curry Milkcap is that we absolutely do not eat it like a standard vegetable.
No, you cannot chop up a basket of candy caps, saute them in butter, and eat them with a fork. It would be a disaster.
That's a total disaster.
The texture is chalky, the flesh can be slightly bitter, and the absolute overwhelming intensity of the Sotolon aroma would make the dish completely inedible. Instead, culinary artists treat this mushroom like a wizard's potion. It is entirely utilized as a high-end spice.
It holds a very rare position in the culinary world. It is treated with the exact same reverence and methodology as a whole, a vanilla bean, a stick of cinnamon, or delicate saffron threads.
Little goes a long way.
Yes. You only require a minuscule fraction of a gram to impart a massive, room-filling flavor profile.
The culinary applications are so fascinating, and honestly, they make me incredibly hungry. The process always starts with desiccation.
Right, the drying process.
The mushrooms must be thoroughly dry to initiate that hydrolysis of quabalactone III. Once dried and brittle, they're usually pulverized into a fine powder. From there, chefs use lipid or liquid extraction.
How so?
They will steep the dried mushroom powder in hot milk or heavy cream, allowing the fat molecules to pull the Sotolon out of the fungal tissue.
That fat extraction is the key to perfectly distributing the intense maple and curry flavor throughout a dessert without ever subjecting the diner to the chalky, brittle texture of the mushroom itself.
Exactly. They strain out the mushroom dust and use that infused milk to create absolute masterpieces. It is heavily utilized in high-end creme brulee, where the burnt sugar notes of the dessert perfectly complement the Sotolon.
Sounds amazing.
It's baked into shortbread cookies, churned into artisanal ice creams, and even used by craft brewers who steep the dried caps in the brewing vats to create rich, complex maple-style beers without adding actual sugar.
Wow.
Mixologists make simple syrups out of it to create woodland-themed cocktails.
And its applications aren't strictly limited to ingestion. One of the more fascinating historical footnotes comes from a 1980 ethnomycological study by Marchand.
What did they find?
They documented that certain individuals utilized dried, crumbled Lactarius camphoratus as a highly aromatic additive in pipe tobacco mixtures.
Imagine sitting around a campfire, smoking a pipe that billows clouds of rich, sweet maple syrup and sharp curry spice. It really speaks to the absolute versatility and power of the aroma.
It certainly does.
And really, if we synthesize everything we've talked about today, the true value of Lactarius camphoratus lies in the multiple perspectives it demands from us.
It is a mirror for whatever scientific discipline is looking at it. To truly comprehend this single organism, you have to examine it through the eyes of an 18th century taxonomist trying to categorize the chaos of nature.
Right.
You need the precision of a flavor chemist decoding the mirror image chirality of molecules. You need the broad view of a forest ecologist mapping the invisible symbiotic internet of tree roots.
Yeah.
You need the targeted focus of a modern pharmacologist searching for the next breakthrough in Alzheimer's treatment. And finally, you need the palate of a pastry chef looking for the ultimate dessert spice.
It really demands all of those lenses to see the whole picture. Which brings us to the end of our deep dive. So, to quickly summarize the wild journey we've been on today.
Get a ride?
We started with a tiny reddish-brown mushroom that 19th century Europeans swore smelled exactly like a bedbug infestation. We tracked the cultural shift as indoor plumbing improved and the scent vocabulary evolved toward curry and maple syrup.
Right.
We watched the mushroom physically shapeshift as the simple act of drying caused water molecules to cleave scentless quabalactone III into the incredibly potent left-handed molecule Sotolon.
We examined the physical traits required to safely identify it, from the chalky stem to the whey-like milk, specifically to avoid toxic lookalikes like Lactarius helvis.
We mapped the violent binary weapon system of its defensive latex, we tracked the devastating hypomyces parasite that hacks its scent factory, and we unpacked an absolute pharmacy of sesquiterpenes and beta-glucans currently being researched to fight cancer and neurodegeneration.
And we learned exactly why eating it turns you into a biological conduit, making your sweat smell like pancakes, fundamentally linking a gourmet ice cream dessert to the tragic reality of maple syrup urine disease.
So to you, the listener, the next time you take a walk in the woods, especially if you find yourself in a quiet, mossy pine forest, I want you to stop and look down at the dirt beneath your boots.
Think about what's really happening down there.
Think about the massive, invisible mycorrhizal internet pumping water to the trees. Think about the binary chemical booby traps waiting to detonate, and the parasites fighting brutal wars over scent factories. It is a wildly complex, highly intelligent chemical world down there.
And I will leave you with a final thought to ponder, based on the absolute cutting edge of modern research. Today, scientists aren't just looking at these mushrooms through microscopes, they are using sophisticated artificial intelligence.
Really? AI?
Yes. Advanced computational models like artificial neural networks, combined with genetic algorithms, along with complex genomic sequencing, are currently being deployed to map the exact step-by-step enzymatic pathways inside these fungi.
Oh, wow.
They are mapping the exact genetic code that dictates how quabalactone III converts into Sotolon, and the exact cellular machinery that assembles those anti-inflammatory sesquiterpenes.
They were literally stealing the blueprints of the chemical factory.
They are. So ask yourself this. As these AI models perfect the blueprints, could we one day entirely isolate and bioengineer these exact fungal pathways in a lab?
Could the future of sustainable plant-based medicine, creating highly complex Alzheimer's drugs or targeted cancer-killing compounds without relying on harsh synthetic chemistry, rely entirely on reverse engineering the biology of a fungus.
Exactly. Could the future of the global flavor manufacturing industry be driven by the genetic code of the Curry Milkcap?
Will the next great medical or culinary breakthrough start with a tiny mushroom that was once despised because it smelled like a bedbug? It's a fascinating question. Thanks for diving deep with us today.